GHMM is like HMM with bigger steps. Instead of labeling every single base separately, it can label whole DNA segments. Before we can predict genes, we must build the GHMM. First, we load the DNA sequence. Next, we define the biological states and how they connect. We then specify transition probabilities and DNA emission probabilities. We also define the start and stop codons that mark gene boundaries. Finally, we convert probabilities into log scores so calculations remain stable. Now the GHMM is ready for Viterbi to begin searching for genes. Now we turn probabilities into scores. First, transition_score checks how likely it is to move from one state to another. For example, moving from intergenic to start may have probability 0.3. We take the log of that probability so the computer can add scores instead of multiplying many tiny numbers. Next, score_segment checks whether a DNA segment fits a state. If the state is start, the segment must be a real start codon like ATG. If the state is stop, it must match a real stop codon like TAA, TAG, or TGA. If it does not match, we give it a huge penalty. For exon and intergenic regions, we score each base one by one. Each base adds evidence, and together they create the total segment score. Finally, length_score checks whether the segment length makes sense. Start and stop must be exactly 3 bases. Other regions are allowed to have different lengths, but lengths far from the expected value get a penalty. So the model asks 3 questions. How likely is the state transition? How well does the DNA segment fit? And is the segment length reasonable? Now we create 2 tables. DP stores the best score for reaching each DNA position. Path stores the actual segment path that produced that score. We start at position 0. The model begins in the intergenic state with score 0 and an empty path. Now comes the main GHMM Viterbi loop. At each position, the model looks at the best states it has already reached. From each previous state, it tries every allowed next state. But unlike a normal HMM, it does not move only one base at a time. It tries possible DNA segments of different lengths. For example, from position I, it may try 1 base, 2 bases, 3 bases, or even longer. Each candidate segment is tested and scored. This is why it is called a generalized HMM. One state can emit a whole segment, not just one nucleotide. Now we score this candidate segment. We start with the best score we've already achieved up to the current position. Then we add the transition score. We add the segment score, which measures how well the DNA fits the state. And we add the length score, which rewards reasonable segment lengths. All 4 pieces of evidence are combined into one total score. The higher the score, the more believable this candidate segment becomes. Many different paths may arrive at the same position and state. But Viterbi doesn't keep them all. It compares the new score with the best score seen so far. If the new path is better, it replaces the old winner. The score is updated, and the entire path is saved. In the end, only the strongest path survives at each position and state. This is what makes Viterbi efficient. It remembers only the best route, not every possible route. The algorithm has now explored all possible segment paths through the DNA. At the final position, several states may still be possible. We compare their scores and choose the highest one. That state becomes the winner. We then retrieve its score and the complete path that produced it. The best scoring path is our final gene prediction. Now it's time to see the answer. We print the original DNA sequence. Then we display the best GHMM path discovered by Viterbi. Each segment is labeled with its predicted biological state. In this example, the model identifies an intergenic region, a start codon, an exon, and a stop codon. The raw DNA has been transformed into a structured gene model. A GHMM works with whole DNA segments instead of one base at a time. It evaluates each segment using transition probabilities, emission probabilities, and length information. Viterbi then searches through all possible segment combinations. At every step, it keeps only the highest scoring path. Together, GHMM and Viterbi convert raw DNA into a predicted gene structure. That's the foundation behind many modern gene-finding algorithms.